All right, well, let's get started. So I want to start with giving you an overview of what we're going to be doing today. Oh, sorry. Oh, already doing a poll.
Sorry, I jumped the gun. Go for it.
Okay, that's all right. All right, so just a brief overview. We're going to start with a poll, and then we're going to go into the demo of JBrowse. And we're going to cover a few topics, mostly about what the data is inside JBrowse. And then we're going to break, take another poll, and after that, we're going to finish with talking about community tracks and also how you can load your own data, your own private data into JBrowse and visualize it in JBrowse. And then we'll finish up with question and answer. So let's start with poll 1. Thank you, Tony. So there are 2 questions. A quick poll. Okay, I think we are reached about 90%. So maybe we can stop the poll. And from the looks of the poll, it looks like most of you have some familiarity, at least you've heard about JBrowse, which is great. And some of you, at least 50% of you have used it and want to learn more, which is fantastic. And so I hope this is really useful to you guys today. All right, let's get started on our demo. So we're going to start off with how to get to JBrowse. So if you're familiar with the TAIR website, when you get to TAIR, there's a top menu bar under which there's an option called Tools. If you click on Tools, you'll see a drop-down menu with several applications, and one of them is JBrowse. You click on it, and the JBrowse window opens. So what are we looking at here? So for the first part, we're going to focus on the top bar, which is at the very top, this whole ruler over here. And we're going to explain what these things are. Now, those of you who have used JBrowse, this is pretty simple. You know what this is, but this is more for the sake of people who are seeing this for the first time. At the very top, you see this, see the ruler at the very top. There's 2 rulers, one at the top and one at the bottom. The one at the top has this red toggle that you can move left and right. And as you move it, you can see that the screen's moving in that same direction. And also, if you see the ruler at the bottom will also be shifting. So we just moved from, I just want to pause for a second. Whoops. Sorry. Whoops. Sorry, we're having some technical issues. Okay, give me just a second. All right. So there are 2 ways in which you can move across the slide. So you can actually slide that red bar either left or right. That will actually move the window quite a lot. And as you slide it, you'll also see the ruler at the bottom sliding. The other way you can move is shorter distance, is by actually clicking on the screen and then moving it left and right. And so that's what you're seeing here, that I'm clicking on the screen here and I'm moving it right and left. So now let's look at the other options. You can also zoom in, you can zoom out. There is these left arrows and right arrows, which can also move left and right. The window right now is focused on chromosome 1, and the position that you're on right now is indicated over here. Now we'll come back to this position when we talk about how you can find your gene of interest. But for the time being, let's keep going. And so now as we slide back, we're going to look at some of the tracks. So each one of these lines that you see here on the right, each one of these tracks are data tracks. And on the left, what you're seeing are the track names that indicate what these tracks are. And at the very top, they have different types. They're categorized in different ways. So what you're seeing at the top is genome reference. So genome reference refers to the assembly. Then in this case, it's TAIR 10. And the assembly of the genome reference is usually grayed out. But if you want to see information on it, you can actually click on the track name and it will pop out information as to the source of the genome and links. But if you also want to confirm that there's actually data behind it, you can actually zoom right in. And if you zoom all the way in, you'll see this very cool graphic where you can actually see all the way right down to the nucleotide level. And now let's zoom out back and let's look at some of the other tracks. So right now I've turned on two other tracks. One is called Araport gene locus and the other is Araport protein coding genes. So let's first look at protein coding genes. So all of this falls under the data set of Araport. This is data that was released by Araport 11 as part of Araport 11. And so what you're looking at right now are the protein coding genes. Again, you can click on the track name and it will give you all the information you need pertaining or the metadata that's associated with each track. You can zoom in, you click on a particular gene, it will give you information on the gene. It will also give you, so it gives a pop-up that gives you a short description of the gene along with other metadata. But you can also click on the gene model. It will also take you to a TAIR locus detail page. This is the TAIR page that is dedicated to this particular gene model in this case. And so now we're gonna close that, this pop-up, and then we'll get back to the gene model track again. And the last option in there is something called view sequence. Now this view sequence option is only available for the gene model tracks. And what you can do with the view sequence option is to be able to manipulate the actual sequence of that particular gene model that we're looking at. And you can do several functions like highlight exons. So when you click on highlight exons, it highlights all the exons in that region in blue. And the other operations you can do include highlighting UTRs. Now you see UTRs highlighted, or highlight introns. You scroll down and you'll see the introns that are going to be highlighted in green. And you can also do other fun things like doing reverse complement of the sequence. You can also add flanking regions to the sequence of interest. So if you want to add, or if you want to pad the gene, pad the sequence with more sequence to its left or right, you can do that. So we just added 1,000 base pairs to the left to upstream of this region. And you can also download the sequence for other analysis. So, and all that is available right from the gene model of whatever gene model of interest you have. So now let's move down to the next track, which is the gene locus track. So for those of you, I'm sure you've recognized that the difference between locus and gene model is the locus is, let's say, AT1G01010, and then the gene model would be the different variations, which is .1, .2, .3. And so now again, when you click on the track name, it gives you the description of it. You can click on the actual locus bar. It will give you, once again, information on the various gene, on that particular locus. And now I want to show you something fun that you can do, which is, so JBrowse is something, though it's running on our server, on your end, it's actually running on your browser, and you have a lot of flexibility in how to modify your view. So any changes you make at your end, don't worry, it's not going to be reflected on our end, and nobody else is going to see it, which is great because we'll come to it when it's time to visualize your private data. But what you can do is, let's say if you don't like the color of the locus track, that salmon color looks not that great, and you want to change it to something that you find appealing, you could you could hit the track name and it gives you an option to edit track configuration. And what you're seeing right here is what's called the JSON that actually codes, that is the code that actually tells how the track should be drawn. So I'm going to edit the salmon and I'm going to convert that into green. And when I say apply, now you see that the track color has been changed to green. Now, this doesn't mean that all other users who are going to come from from this time onwards are going to see the same track or same color. Once you refresh this browser, you're going to see that it's going to go back to the default, which is salmon. Okay, so that's as far as locus is concerned. Where are we? Okay, so now let's turn on. So we looked at gene models and locus of Rapport 11. Now let's turn on the ones from the previous version, which is TAIR 10. And why would we do something like this? So let's say, for example, if you have a gene of interest and you want to know with a new version of the genome, what's happened to your locus? Has there been a new gene model that's been identified? So how would you do that? So the way you would do that is you would go up to, remember where we had the chromosomal position, you would go up to that and you would enter in the name of your locus. And from that position, as you enter in the name of your locus, it automatically pulls down the various gene models, the locus name, and the gene models associated with it. So right now we're interested in the locus. So we select the locus. It gives you all the positions in JBrowse that has the locus in it. And then now you see that, so we're going to zoom out a little bit so you can see it much clearer. You can see that the window is now centered on that particular locus that you were interested in seeing. For example, there's the locus that you were interested in seeing. And now what you're seeing is that the information on that locus on Rapport 11, as well as in TAIR 10, both the locus information and the protein coding genes. Now here is a point where something called highlighting comes in very useful. So there's an option to highlight a region. So if you want to really focus on one gene or one area, you can actually set a highlighter by selecting that region. And by having that highlight, you can actually makes it much easier to see visually what's going on. So you can see clearly here that we can make a visual comparison, and you can see that there is, in the case of TAIR 10, this particular locus had one gene model, whereas in the case of Rapport 11, this particular locus now has two gene models identified. So there is one immediate comparison that you can make right there. Let me just look at my notes here for what we're coming next. All right, so now, oh yeah, so this is going to show you more information. You can actually zoom in and look at the gene model, the new gene model, and understand what that change, what the reason for that change is. Okay, so now let's fast forward a little bit. Okay, so now we're gonna close the Rapport data section and we're gonna look at the section called community data. So what is community data? The community data refers to a whole set of tracks that contain data that's been submitted to us by you, the scientists, you, the users. So this is published data or data that's been submitted for publishing. And we have categorized them on the left as to the type of data set it is or what it indicates. For example, we have data on variation data, which is tDNA-seq data here. And you can zoom in on here. So depending on the type of data set and the metadata that the user provides us, we can customize the track to fit the view that you want, that you, the user who's submitting the data, want to see it as. So, oops, again, excuse me. So let's go on. All right. So when you're gonna, so let's have a look at tDNA-seq data. You can zoom in and you can right click. So there are two clicks that you can do. You can actually click on the track information and it will take you to a link or information like this. Or, so for example, this is actually the track data, the information associated with the track. And what you can see here is the name of the track, the description, the source of the track, and the URL for the data source. So let's keep going here. And so now you can right click on that particular locus and it will give you various options, including the detail page for that particular tDNA within TAIR. So when I'm going to click on it now, it's going to open a page showing you the polymorphism page within TAIR for that particular locus. I'm going to fast forward it a little bit because there's a little bit of redundancy here. And there are other data sets also available. There are repeat sequences with tandem repeats. Running a little short of time, so I'm just going to forward it a little bit. But now I'm going to get to the point. So feel free to actually view any of these data sets. You can actually submit your data and you provide us with the metadata associated with it, and we can have it visualized the way you want. Now, what is the advantage of doing something like this? For example, what does it mean to you if with the data sets that are already there? So what you're looking at here on the left are data that's from a study on hypoxia. And this is ATAC-seq data done on control and on hypoxia data sets. And so what we're going to do now is compare between controls and hypoxia data sets. And you see the ATAC-seq data now being mapped. You have control at the top, and then you have the hypoxia data set at the bottom. Now, visually, you can see that there is some difference, but there is one way that you can compare the two, the control versus the hypoxia, is to actually drag your mouse over it and actually compare the values visually, but that's pretty cumbersome. So how can you actually compare the two in a meaningful way and be able to see if there is any real difference? Well, there's a really cool function in JBrowse called combination tracks. So if you go up to the genome, the track menu up the top, there's an option called add combination track. If you click on that, it's going to open up a blank track after these two tracks called combination track. And you drag the two tracks that you want to compare into this combination track. And now when you drag in the second one, it's going to give you an option. Do you want to edit? Do you want to add the two of them? Do you want to subtract? And obviously, since we're comparing, we want to subtract. So we're going to click subtract. And then we're going to ask to combine the tracks. And when it combines the tracks, you're going to see something really cool. You see that the differences between the two tracks are now indicated really clearly. What you're seeing in the combination tracks, for every position, what is the difference in the reading between the two tracks? Okay, so that's combination tracks. The other feature that we have is we don't just have data that you can, we don't have just visualization tools for data that's posted into JBrowse, but also if you have data posted in other repositories, like for example, Koji, you can also provide us the metadata on that and we can link it up. What you're seeing here is a menu that users can come in and select the type of data set they want to see. The metadata that we want from you is basically, similar to what's listed here. Basically, the ID of the experiment in Koji and the name of the experiment and so on and so forth. These details, if you provide to us, we can then populate this for any of your users who want to see your data that's already in Koji, so that we don't have to replicate that again over here. I'm going to fast forward this at this point, and I'm going to move forward. So this is just showing you some of the how Koji data actually looks on JBrowse. So let's take a quick break here and go on to the second poll. I apologize, I'm running a little over time, but let's do the quick poll. And so as I said at the beginning, if we don't have time for questions, or if we don't have time to get to all your questions, don't worry. Please post them on the chat, and I'll reply to them by email. Okay. Still getting. Okay, I think that's good. We have 87% voted. So most people haven't worked on Arblausis Thaliana, and as far as data sets, there's quite a diversity. That's great. Okay, so let's go on ahead to the next stage, which is actually, I think, to me, the most important part of today's demo. And that is how to load and view your own private data in JBrowse. As I said, JBrowse has two instances. There's the instance that's running on our server, but also the instance that's running on your browser. So you can actually load your private data on your viewer and then view it without sharing it with anyone. So it's not just that you can only load published data on here. So what I'm doing right now here is let's say I have a genome that has nothing to do with Arabidopsis, and I can load my own genome into the JBrowse at TAIR. Don't worry, we're not going to see it. Nobody else is going to see it. Only you're going to see it, and you're going to see it only in that particular web session. So here I'm scrolling through and I'm finding a file, a genome file, a FASTA file of a genome sequence. and I'm loading it in there. And once I load it in there, it's going to appear the same way you see the reference genome track of Arabidopsis. So you can load. I don't think I'm loading it in this video, but just to show you how you can do it. So you can load your own genome if it's different than Arabidopsis. You can also load your own experimental data. Let's say, for example, if you have some data that's not yet been published, but you want to see what that data would look like when it's published, and or you want to share it with your, or you want to see what the data looks like, you want to do some comparative analysis, you can load your own data sets on your private instance of JBrowse. And this is how you do it. Under track, you go to open track file, zoom out, and now you can select the file that you want to load. So I'm going to load a big file, which is, give me just 1 second. I think it's a TrapSeq data set. And I'm going to open that TrapSeq data set. You can name it anything you want. And now when you click on it and you zoom out, you're going to see that TrapSeq data showing up on your browser. Now this will only be visible to you. And so this is a great way for you to see what your private data looks like on JBrowse, and that eventually when you publish, you can actually then be able to see it. So when you look at the track information, you'll see something called local tracks. So when you see something called local tracks, that means you know that it's only available for that particular session. And you can do the other fun things, like you can change the color on this one just like you change with the other ones. So that's great. It's a fun thing to do. And let me see, let me make sure that I'm on the. Okay, so we have 8 minutes left. Let me just quickly go through 2 other data sets we have, and then we will close up. Second, please. All right. So the other 2 data sets that I haven't talked about today are these 2 data sets called VistaPlots and Phytosome 13. These are both data sets that we got from Phytosome. And so those of you who are familiar with VistaPlots know what they are. They're basically pairwise alignment comparison between a genome-wide comparison between Arabidopsis thaliana and these various other genomes. And they're plotted in a way that you can actually tell the difference. They have different colors for different regions. For example, UTRs are colored differently from exons. And so you can do this comparison between different species, and you can map as mapped in comparison to Arabidopsis. And again, this is real-time data from Phytosome. So fast forward a little bit here. And you can actually click on the track and it will take you to Phytosome for more information on those tracks and additional information that you want related to that locus. The other data set we have is Phytosome 13. So if you go to Phytosome website right now, the latest version that's been released is Phytosome 12. Phytosome 13 is still in beta, but they've given us permission to actually make that data available. And so if you go to our website, you can see Phytosome 13 data now organized in different categories. And you can select that data and now you can visualize it. And in the same way as the other tracks, you can actually click on them and you can change whatever information you want. You can actually, when you click on the actual track, you get information about, and also you can go to the Phytosome detail page on about that locus. So we're going to click on that. It's going to show you what that gene looks like in Phytosome and gene information and information associated with that gene. Okay, that was. I apologize for rushing through it at the very end. I just want to say one more thing before we finish up. If you're interested in sending us data, these are the kind of input formats that work. If you want to send us annotations, send them to us in GFF, BED files, or as GenPack files. The types of experimental data that we are supporting so far, and the list keeps on increasing. So RNA-seq, ChIP-seq, ATAC-seq, TrapSeq, methylation data. We're in the process of implementing additional methylation data. We're going to add those into it. And as far as experimental data is concerned, when you send it to us, please send it to us either as BAM files or BigWig files. If you have any more inquiries, please email us at info@arabidopsis.org. It's very important to acknowledge all the people who helped a lot in making this happen. People who helped us build the JBrowse instance, as well as people who have submitted data, and of course all the subscribers who support TAIR. And this is the awesome Phoenix staff. Those of you who have attended the webinars before, you got a chance to meet Lenore. If you attend future webinars, you'll get to meet Tanya as well. And here are the other webinars that we've had. The first one we've already had it, but the recorded thing of that will be available soon. Upcoming, we're going to have Go Annotations and TAIR Go Tools by Tanya. That's coming up June 19th and the 23rd, and as well as Gene Functional Data by Lenore on 26th and the 30th. And if you want more information on the upcoming webinars, you can go to that link. Okay, so with 3 minutes left, let's go to Q&A.
All right, we have a number of questions here, and I'll try to go through them one by one. So the first question was, what's the difference between the black protein coding genes and pink gene locus horizontal bars?
I'm sorry, black protein coding genes and what?
And the pink gene locus bars.
Oh, okay. So the gene locus is the main locus, which has, so the gene. Do you mean the difference between the gene models and the locus? Is that the question?
I think so.
Okay. So the difference between gene models and the question is, for every locus, you can have multiple gene models, and so that's essentially the difference. So, for example, if you have a locus like AT1G01010, you can have multiple gene models like 0.1, 0.2, 0.3, and so forth. And of course, it has a representative gene model as well.
Okay, so and then a question about how you search for a specific gene using a keyword or terms in JBrowse. Can you do that?
Yeah, so I'm not sure if you saw what I did earlier. You can actually go to that chromosome position where it had the CHR1, 2, so on and so forth. And you can actually, let me see if I can pull that up here. So you can actually go to this place and you can enter the gene information, and it'll actually pull up that gene on the viewer itself.
Okay. And then a question, several people were wondering about looking at different genome assemblies. So yeah. People who are interested in looking at tomato and Medicago, Solanaceae. So how could you look at different assemblies?
Yeah, so right now you can do that on your own private instance. For example, you can use the technique I was telling you about, where you can load your own genome on the browser and see it. We are in the process of adding on other genomes that are relevant to people that work on Arabidopsis as well. So if you can think of species that would be relevant comparisons to Arabidopsis that would make sense for us to add, please let us know, and we can add those genomes. If there's enough demand for it, if there's enough value to it, we'll definitely add those as set genomes along with the Arabidopsis thaliana genome. But for your need, you can actually do that right now. This is the same way I showed you. You can load your own genome and add data sets. But as I said, it's temporary, it's transient. The one thing I kind of skipped through was there is a feature at the very top called share. So this is great because if you want to share the actual screenshot that you're looking at, you don't need to print it or take screenshots or anything. That when you click on share, it'll actually give you the entire link. And so if you have collaborators or if you want to send your data to reviewers, you can actually send that link with the exact view that you want them to see, and they'll be able to see it.
And a related question to that was whether the private view required registration. So the question was that if you could save it, and it's clear that you can save the tracks, but does it require registration?
No, it does not.
Okay. I think it's 8:30 now, so there's a few other questions that maybe Shabri can respond to people directly.
I will do. Will do. Thank you so much for joining us, everybody.